Now we're going to continue on with our topics in cells, and we're going to look at cell transport. Cell transport is how a particular cell brings in and takes out things that it either wants or needs to get rid of. How is it able to do that? We're going to categorize cell transport into two categories. One is passive transport, which happens without energy, and the other is active transport, which requires energy in order to achieve it. Before we can really get into transport, we need to think about how would something move through the cell membrane. So remember that the membrane is made of those phospholipids with their hydrophilic heads and their hydrophobic tails. And so some things can move through the membrane without any problem, but other things cannot. Small molecules can move through the cell membrane pretty easily, but large molecules, even as large as a sugar molecule, which is still pretty small, does not have the ability to move through. Water can move through the membrane, but other things cannot. And so this selective permeability gives the cell the ability to allow some things in and other things not in. One good way to think about selective permeability would be like going to Disneyland. Okay, you want to get through the gate at Disneyland. There's a few things that needs to be true. Number one, you need to have a ticket. Great. Okay, because you're going to have to fit through these little openings. So you're going to need to have a ticket, right? Number two, you're going to need to pass their safety screening. So they're going to take you through a safety screening. I bet the next time you go to Disneyland, at that exact same safety screening where they check your bags and they make sure there's no weapons in there, they're also going to take your temperature. So they're going to do a health screening as well. You're going to go through all of these screenings, and if you pass all of these screenings, then you will be allowed into Disneyland. That is selective permeability. Sure, there's a way to get through, but you have to follow these particular protocol, and only some people are allowed through. If you have a weapon, if you have a fever, if you don't have a ticket, you are not permitted through. So that's what selective permeability really means. Movement of water in molecules is based off of concentration. Concentration is looking at how much of the solute is dissolved in the solvent. We talked about solutions earlier in the semester, but as a reminder, the solute is the thing that is dissolved and the solvent is the thing that does the dissolving. So remember, making Kool-Aid, the solute is the powder itself and the solvent is the water that you're mixing it in. When we see two different solutions with a selectively permeable membrane between them, what we notice is that our chemicals want more space. The chemicals would like to move from a concentrated or crowded area to a more dilute area. That's what they would like to do. The water feels the same way. It also wants to move from an area of crowded to an area of space, of dilute. So the water wants to move away from the water and the solute wants to move away from the solute. And so we seem to get this kind of balance. Keeping the water balance, maintaining it and then keeping it is one of the most important homeostasis events that happens in our body. If you drink plain water, we want that water to come into our cells, but we don't want it to flood into our cells and burst them and break them open. Okay, we want it to come in gently. We want it to come in where it's supposed to go. We want to keep our hydration. We also want to keep our salt balance. So we want this increase in hydration, but we need to keep the balance. So homeostasis, remember, is this dynamic balance where you have a shift and then you have a return and a shift and a return. And so we're going to be looking at how our cells maintain this homeostasis inside of our cells. So as I mentioned before, there are two classifications for transport. The first one is passive. Passive transport does not require ATP. And so what you're going to notice is that in each one of these examples, passive transport means that we are going along our concentration gradient. Okay, so we're going to look at what a concentration gradient might look like. This is what a concentration gradient may look like. So in a concentration gradient, we have one area that is concentrated and another area that is dilute. And what we said was the molecules themselves would like to move down the concentration gradient. So the solute wants to move this way. The water is more concentrated over here. The water molecules are more concentrated where the solute is dilute. I think that's important enough and possibly confusing enough that I want to write it down. The water is more concentrated where the solute is dilute. So for the water to move down its concentration gradient, the water is going to want to come from over here and go this way. And the particles, the solute, is going to want to go that way. So both of these want to be working in opposite directions. And so what we see is that diffusion is the movement of molecules along their concentration gradient. Diffusion of the solute. Water is the movement of water along its concentration gradient. That diffusion of water is called osmosis, and it is typically the opposite direction of the diffusion of the solute. So if water wants to go this way, solute wants to go this way. So osmosis and diffusion work in opposite directions because they're moving different particles. Diffusion that just happens straight through the cell membrane is just called diffusion. If that particle is too big to get through the membrane on its own, it may go through a port. And if it does that, then we call it facilitated diffusion. It just means that it needs a tunnel or a path in order to diffuse, but it is still going along its concentration gradient. So what's really important that you notice is that passive transport has no ATP, and that's because everything is moving along their specific gradients. Active transport is going to be moving molecules against their concentration gradient. And because we're going against the concentration gradient, this requires ATP. So this is a cell membrane. These purple things are the phospholipids, and this blue thing is a protein. What we see in active transport as we move against the concentration gradient, there would be a large concentration on one side of the protein. And so we know based on diffusion that these molecules want to move in, but instead we're going to move this molecule out against its concentration gradient. It is not going to want to go there. It's going to be kicking and screaming. So we're going to need to shove it through, okay? We're going to need to force it across. And the way that we do that is by spending some of that cellular energy we've been talking about of ATP. We're going to use ATP. When we use ATP, we turn it into something called ADP, which for now you can just write down, and we'll explain more later. And as we use up ATP, we are able to get enough energy to shove that molecule against its concentration gradient and out of the cell. So this is usually through a protein, and it's the movement of a molecule against its concentration gradient. If we need to move something that's way too big, we're going to use something called endocytosis or exocytosis in order to move that thing. And we're actually going to spend a whole slide just on that topic in just a moment. So for now, endocytosis is when we're moving something too big to fit through the membrane. Instead, we're going to need to come up with a whole different pathway in order to move it. So here we're spending just a little bit more time looking at osmosis versus diffusion. So remember that osmosis is the movement of water. This is going to move the water from where the water is concentrated to where the water is dilute. So if you look at this glass, the water is more concentrated on this side, and the water is less concentrated on that side, which means that our water wants to be moving from this side of the glass over here. That's where the water wants to go. the particles, the little blue spheres, the sugar molecules, are more concentrated over here. And they're going to want to move across the membrane this way over to this more dilute side. So once again, we see the water wanting to go one way and the solute wanting to go the opposite direction. So the water and the solute are going to try to go in opposite directions in order to establish equilibrium. Equilibrium is maintained through homeostasis. So first we establish an equilibrium with proper concentrations, and then we maintain that equilibrium through homeostasis. We can see osmosis by putting cells into different solutions and watching what happens. So if we take a cell, this is a red blood cell, and red blood cells kind of look like little donuts a little bit. So red blood cells, they're like, or inner tubes, they're fatter on the outside and they're skinny in the middle. And so what happens is if we put red blood cells in an isotonic solution, isotonic means that it's the same concentration on the outside as we have on the inside of the cell. Isotonic, it's stable. Then what we see is the cell stays stable. It looks just like a red blood cell. If we put the cell into a hypotonic solution, think like hypotonic, it gets fat. So in a hypotonic solution, we have a high concentration of water on the outside of the cell. So we have very little solute in the liquid. And so all of the water, so imagine you're a cell, you're a red blood cell. You have a certain number of molecules concentrated inside. If you put this cell in just plain water with no solute in the water, all the water is going to want to come rushing in. And the cell is going to swell up and get really fat. This is a hypotonic solution. Hypotonic, that big O, right? Hypertonic means that in a hypertonic solution, we actually have way more solute on the outside. So you'll have to bear with me because I'm not going to count, but just imagine. In a hypertonic solution, we have way more solute on the outside. And so the water wants to leave the cell to come follow the solute out. And so that leaves the cell looking all kind of shriveled up and broken down. That's a hypertonic solution. And remember, an isotonic solution would be really hard for me to draw because it would have to have the exact same number of solute molecules on the inside as on the outside. It would have to be exactly the same concentration. So I'm not going to try to draw an isotonic solution, but just know that the water balance moving in and out would be exactly the same. So you'd have the same number of water molecules wanting to leave the cell as wanting to come into the cell, and the cell would stay nice and balanced in an isotonic solution. The last kind of transport topic that we have to talk about is endocytosis and exocytosis. And so in endocytosis, we are bringing, like, think endo, enter, okay? We're bringing particles into the cell. And in exocytosis, we are expelling them out of the cell. And so we kind of looked at exocytosis already when we talked about the concept of the secretory vesicle. So this might be material that we need to get rid of. It could be proteins that we made that we want to get rid of, or it could be an old virus that came in and we were able to break it down and now we just want to get rid of it. In both of these pictures, we see this vesicle that's coming up to the edge of the plasma membrane. And because the plasma membrane is all made of the same stuff, when they touch, they merge. Think of like slime or silly putty or Play-Doh. As you bring them together and they touch together, then they're going to merge together into one continuous piece. And so this vesicle comes up to the surface of the cell and it touches, and then it all becomes the same material. And so it kind of opens up and it allows this secretion to get removed. So that is exocytosis. So cytosis is, cyto means cell. So it's exiting the cell, okay, exocytosis. Endocytosis is the opposite. So this is where we're bringing in this particular case, we might be bringing in a particular molecule. We might be bringing in some liquid in the extracellular fluid, or we might be bringing in a particular chunk of food that was kind of floating by that we want to bring into the cell. And so if we want to bring in food, if it's a solid, we call that phagocytosis, where we bring in a phage, like a thing, okay? And if we are bringing in a liquid, we call it pinocytosis. And so that brings that liquid into the cell. And so we put them into vesicles, and then you can imagine the lysosome would come and connect to the vesicle then, and it would digest the material that was brought in, and then we would distribute that within the cell where it was necessary. So if something's too big to go through the cell membrane, then we use endocytosis or exocytosis in order to transport it.